Hello, this is Tommy. Welcome back to Chatomics. In today's video, I'm going to show you how to use Bioconductor to create a genomic ranges object using only the canonical transcripts. For each gene in the genome, there could be actually multiple transcripts for the same gene, or the isoforms for the same gene. And there are different transcript reference systems that are available in the public domain. For example, this is UCSC known gene, this from the UCSC genome browser. And they actually aggregate those gene predictions using its own transcript IDs. Many of them overlap with, for example, RefSeq and also Ensemble. Or you have this system called MAIN, which stands for Matched Annotation from NCBI and EMBL-EBI. For example, the RefSeq uses, they are curated by NCBI and focus on high confidence and experimentally validated transcripts. And why does it matter, right? In practice, it matters because if you use different isoforms, and when you annotate, for example, a variants, the positions for that transcripts can be different. And also even the amino acid may actually different depending on which transcript you use to annotate your variants. And for example, another example is that when you annotate peaks, which for the ChIP-seq data, and it binds to the genome, and you want to annotate a peak to that peak to the closest gene. And depending on which transcript you use, the annotation may be different. And what do I mean actually? So we can go to IGV and take a look. For example, I'm just looking at the TP53 locus, and this is RefSeq selected. There's only actually single transcripts for TP53 or the canonical transcripts. This is the exons, those are the introns. And you transcribe from this direction to this direction. But if you go to Files, Load from Server, and click the annotations, in the genes here, you can select other annotations. For example, RefSeq curated, or the other stuff that, for example, gene code or main annotations. I select this one and loaded it, and you will see this is the RefSeq curated. And in this case, you have so many different isoforms or transcripts for the same gene. They also may have different transcriptional start site. For example, in here, this is the transcriptional start site. And for those transcripts, the transcriptional start site is here. And here is just some example of ChIP-seq track. In this case, a YAP task and tab for binding sites. And if you use this transcript as the annotation, and you see this peaks, although it's binding to the promoter region or the transcription start region. But if you use those transcripts, those will be annotated as distal upstream enhancer peaks. You'll see why, if you choose different isoforms, it can actually affect your downstream analysis or conclusions even. And luckily, we can choose only one single script for all the canonical ones from each gene. I'm going to show you how to do that within Bioconductor. Let's go back to R, and we'll load those libraries. Ranges, so annotation package, annotation hub, and ensemble DB. By the way, I will have the R Markdown file and the blog link in the description of this video so you can follow along. We'll actually download this ensemble database. And if you look at this is the ensemble database here. It's created by the ensemble DB package. And we can actually create the transcripts using the transcripts function from the genomic ranges package. And you supply the database into it. This is the transcripts. And each row here will be one single transcript. And you see this is the transcript ID, this is chromosome name, start, end. And then you also have the For example, both of them are from the same gene DDX11L1, but those are two different isoforms. And you have another column here that is called TX is canonical, which is very handy. If it's 0, means it's not canonical, and if it's 1, then that is canonical. We can use that to filter the transcripts. But if you look at the seq names or the chromosomes, if you look at them, you have chromosome 1 to 10 to 20, 10, 19 here, but you also have this kind of weird chromosome names. You want to get rid of them, for example. You can use keep standard chromosomes, pooling mode equals course to do that. And then if you look at the seq names, now you get rid of those weird names. You only have 1 to 20, and then 23, 22 here, then mitochondria, X chromosome, and the Y chromosome. We also want to somehow rename those to CHR1, CHR10 instead of just the numbers. Usually the ensemble annotation, they don't have the chromosome prefix. If you look at the seq levels here, this 1 to X and Y here, we're going to actually create this new seq levels to chromosome 1 to just add the prefix, and we can add the new seq names as a named vector, and now we can change it, use the rename seq levels. Now the transcripts, now they have the CHR prefix. We also want to add another column as a symbol name, or the gene symbols, although we have this TX external name here, but this also has these numbers here to indicate which transcript it is. We can use the map ID function, and then we add another column here called symbol. And the key will be the transcript ID, which is this column. And then we want to return the symbol. The key type will be TXTB. If we do that, now let's take a look again. Now in the last column, we have the symbol. And we also have the canonical transcript. That's how you can actually get a genomic ranges object with only the canonical transcripts. It can be actually really handy sometimes. And you can use this to annotate your ChIP-seq peaks, for example, using the ChIP-seeker package. Okay. In my next blog post, I will show you how to calculate regulatory potential scores using the ChIP-seq peaks and this annotation file, this canonical transcript annotation file. I will see you next time. Happy learning, and stay tuned for my next video.